Okay, so hello everyone, and I have the pleasure to introduce Sawadi. You know Sawadi, Fransisco Ndunguru, who is a PhD student, actually not in this doctoral school, but in the agricultural doctoral school, and we co-supervise him with Tanszék. Sawadi is from Tanzania, and he arrived in Debreceni in 2020, probably in the midst of the second or the third wave of the pandemic, and so he spent one wonderful week in the Aranyvika before moving here closer to us on campus, and then he has been doing some quite interesting work on Japanese quail and some molecular mechanisms influencing life history traits in Japanese quails. And I must admit that some of the results I find very intriguing and very interesting, and this is some of it that he will talk about today. Not all of it, because he's also involved in different experiments, but a subset of his work that he's going to present today. So, Sawadi, please.
Yeah, thank you very much, Dr. Prada. As he has been introduced, that I'm working together with the Professor Levente and him. So actually we are working on amino acids, how do they influence growth and aging. So today I'm going to present some of the results that we have found in our studies, and specifically in Japanese quail. And we know how mothers actually love us, and then during our embryonic development in the womb, they have to allocate some nutrients for us during the growth in order to grow well after we are born. And during that early stage of development, it's important to understand that all those phenotypic variations that occur during birth are related to the allocation of the nutrients during the embryonic development. So embryonic stages are very important. Mothers have to allocate all the necessary requirements, including nutrients, vitamins, and all other stuffs that are important in post-natal development. So embryonic development is one of the critical stages in development, which influences the offspring's development in later stages. Basically, are important in the physiological responses. So embryos actually incorporate the maternal signals from the mothers that they acquired for adaptive development in later stages. So the allocation of the materials from the mother to the embryo development, to the embryo that is developing, depends on several factors. One is either the type of quality, the type and the quality of diet, the type and the quality of diet, or the predation risk or social environments. For example, if you check in birds, you will find that if the habitat is not good enough, the bird will not be able to inhabit that area and hence to nesting. And then if maybe food availability in that area is not enough, that means the birds cannot select that area for nesting in order to have babies. And also, if you find that there are predation risks in that area, that the organisms or birds sense that this area is not potential for nesting, it's not potential to lay the eggs. It will not necessarily lay the eggs in that area because there is a probability that the eggs will be predated and then they will not be having the babies anymore. So they don't allocate much more resources to the eggs in such a way that they don't lay much more eggs. And then the social environments here is how they are connected to the other birds, for example. How do we live in the environment? How the birds are connected, they are socializing with the other birds. If we live in a social environment which is much more connected to others, that means we become happy. Even the allocation of the nutrients, the hormones, the eggs are becoming very potential to these eggs because they are happy in that area and they are comfortable in that case. They are able to allocate the necessary nutrients and all the resources to the eggs so that they can influence the development of the embryos in the egg. So one of the important resources that birds allocate to the eggs is the hormones, in which hormones are influencing the growth and development of the embryos. And one of the systems that is very important is this insulin signaling pathways, which senses the status of the nutrients and regulate metabolism, growth, and development in birds. In vertebrates, this IGF-1 is connected to this system, IS system, the insulin signaling system, which is important also in cell proliferation, migration of the differentiation of cells, and protein synthesis, which is important in growth and development of the eggs. Despite of this fundamental importance of the insulin signaling pathways, this has not been yet studied to its maximum in this species, in bird species. So ISS is being connected to other pathways. It does not work in its isolation. It has got the influence in other pathways like this mechanistic target of rapamycin. They work together. This is the most influential pathways. That is the major intracellular pathways, which is connected to nutrients. It senses all types of nutrients, and it's a protein kinase, which regulates other activities, including the metabolism, catabolic and anabolic activities in the presence of nutrients. So, sorry, I got back. Who discovered this mTOR pathway? There are prominent scientists who have been dealing with this mTOR pathway. They wanted to understand how these pathways works in different organisms. How did they discover? There are 6 scientists here that have been mentioned. One is Fransisco, who is regarded as the father of rapamycin, and Abraham is here, Sabatin, Stuart Scriber, and Michael Hall, and Levy. These are the prominent scientists who discovered this mTOR pathway that senses the nutrients. How did it begin? It began with this place that the prominent microbiologist, Georges Nogrit, he was working in this area, the East Island in Chile. This is South America. This island is also called Rapa Nui. He collected the soil samples after found that people in this area are walking in a barefoot, but they don't get this disease tetanus called it, but it's caused by Clostridium tetani, the kind of bacteria. But he was surprised why these people are walking barefoot, but still they don't get this tetanus, because this area is much more occupied by horses, and the areas where horses are available, they are much more contaminated with this Clostridium tetani, which causes the tetanus. So he was curious enough to understand what's happening in this area, what's happening to these people, why don't they get this tetanus? So he collected the soil samples to investigate what's the problem with this area, or what's so funny, but he didn't find anything in this soil. And this island, actually, the prominent feature in this island is the presence of this statue. This kind of monuments, I don't know what to call it, but this is like a prominent feature. So after that, he didn't find anything, he didn't stop there. He went further and sent the soil samples to Dr. Sigo. He was working in IST Pharmaceuticals, recently it's called Pfizer. Those developed the coronavirus, I don't know. So Sego was working with this company, the pharmaceutical company, and he investigated what is available in the soil samples. He found that there is a compound which is called the rapamycin. He named it rapamycin in the name of the origin of the place, Rapa Nui. So this compound was found to be antifungal activity. that it had got implication in antifungal prevention. One of the diseases that is Candida albicans, so they published this paper in 1975 to indicate that the compound that was in soil is called rapamycin, and it's important in antifungal activity. Then it prevents this bacteria that is causing tetanus to develop in that area. That's why people are working berry in food, but they don't get tetanus because of this, the presence of this compound. So it kills those kind of bacteria. But rapamycin doesn't only have this antifungal activity. It also has some major important activity. It's used as some immunosuppressant that prevents the organ transplant rejection. Some people, if you have a failure in, let's say, kidney, and then they have to take another kidney from one person, then implant it into your body. So there is a probability that immune system of your body will reject that kidney that has been implanted in you. So to avoid that, they use a drug. But initially, the F56 is an immune that is preventing the rejection of the organ. But later, they discovered this rapamycin, which was a replacement to organ transplant rejection as a drug in medicine. So it is also used in cancer treatment and also cardiovascular disease treatment. What the discovery of this during the discovery, there was a lot of stages in discovery of this mTOR pathway. In 1990, the scientist Scriba found rapamycin is working, or it is attaching to the same place as F56, which is the immune and anti-immune that is preventing the rejection. So they attach to the same place. And again, Sawadi found the same target where rapamycin is binding this protein, FB binding protein 17. But at the same year, Scriba again found that the same kind of rapamycin is in connection with the FK50, which is anti-immune system, and also Abraham found the same with these geneticists who found that there is a gene which is connected to the target cell cycle that is involved in the cell cycle arrest. And they named this finally as mechanistic target of rapamycin, where this protein gene is binding, this rapamycin is binding to this kind of protein called the target of rapamycin. So rapamycin was binding to the specific protein, where they called this as mechanistic target of rapamycin. So this rapamycin is actually a very complex network. It's a pathway that is implicated to the pathway of which there is a complex network of this rapamycin, but it senses a lot of things. But one cannot understand it in a quick way. So there are a lot of diagrams that are related to this network. You can just search it to this mTOR signaling. You will find a lot of diagrams related to this. But you can take this in a simple way, that mTOR is composed of these two components, but they differ in the proteins that are scaffolding proteins that are connected to it. But they sense a lot of things, apart from just nutrients, but there are other growth factors, hypoxia, that is oxygen, stress, and amino acids. But the amino acid doesn't connect to the same pathway as the other factors. But amino acid has got its own pathway where it connects to mTOR C1, the complex 1 and complex 2. But in turn, what the end product is protein synthesis, ribosomal synthesis, or survival, growth, and the other stuff that are connected to mTOR pathway. But the mTOR has got other upstream factors like the serine 6 kinases, but it is connected to mTOR C1. Finally, we get growth or either production and other biological functions. So, being connected to rapamycin being inhibiting this mTOR pathways is binding to this protein. So all stimulations that are coming from these cues, they go to mTOR pathway. But if you inject rapamycin to that organism, rapamycin is inhibited. It's not stimulated by these pathways. Otherwise, stress just inhibits mTOR, but rapamycin also inhibits mTOR. But if stress, all these factors are available, and then you inject rapamycin to the organism, mTOR pathway is inhibited, then it stimulates the autophagy, which is the clearing of the cell organism. Once cellular components are being used, then you can clear this by using, remove by using this process, the autophagy. But if mTOR pathway is activated, it influences growth, metabolism, cellular proliferation, and the macronutrient biogenesis, like protein synthesis, lipid synthesis, and other metabolic pathways that are related to this. But mTOR, not necessarily that it has some influence in growth, but it causes some diseases that is connected to nutrient. Once this is the nutrients that are increasing in the body, immune system is declining. There is a cause of diabetes, autoimmune disease, can develop heart diseases and other related diseases that are connected to aging. If you have got these diseases, this is particularly that it's open that you might be, your age might increase significantly. So why this mTOR is connected to this disease? Because it diminishes or it reduces the immune response systems. So you can get these cardiovascular diseases and finally cancer because of multiple cellular divisions that are connected to nutrient availability, because mTOR pathway is on, mTOR pathway is activated because of the presence of nutrients. And so, how do we prevent this? We have to inhibit the activation of the mTOR pathway. How do we inhibit the activation of mTOR pathway to influence autophagy, the clearance of the usually cellular materials? There are cellular materials that have to be removed. They look like a trash inside the cell. So how to remove this is by autophagy. How do we activate autophagy? It's just by reducing the activity of mTOR pathway. We have to stop mTOR pathway. How do we stop mTOR pathway? It's just to reduce the amount of nutrients that we have to eat. So we extend our lives. Lives start with a single cell. So the bigger organisms that they need much more food in order to grow, and they live longer than the smaller organism as compared to, you can see here, this is flies, but they live shorter lifespan as compared to the multicellular media organisms. These multicellular organisms, they usually need a lot of nutrients, and they need to stimulate more their mTOR pathway. But within species, there is a difference. Bigger animals in these flies, they usually live shorter lifespan as compared to small animals. The bigger dog actually lives shorter lifespan compared to the small dog. So in this case, more nutrients are required for bigger animals, but within the species itself, if it's big, then it will live shorter lifespan as compared to the small animal in the same species. So nutrients are important, at least to increase the body size. So if we need to extend our lifespan, we need to reduce the amount of nutrients that we eat in order to become more healthy. This pathway actually is conserved in most of the animal taxa. So you can see here from yeast. Then if we do some kind of dust restriction, the signals come to top, and then also worms, flies, and mammals that are connected signals. So if you do that, some kind of dust restriction, then mTOR is inhibited, no further activation of the mTOR, no further diseases, no further aging. So that means you increase your lifespan. So we need to live more life in this case, then we have to reduce the amount of nutrients that we need to eat. If you check in the old life, we could say that not all mTOR activation is bad. It depends on how old you are, the stage of life that you are. For the old people, if you are the elder, then you need activation of the mTOR. Why do you need activation of the mTOR? Actually, during protein synthesis stage, at the younger stage, there is high metabolic pathways that is related to protein synthesis as compared to the older people. So if you are food insecure, that you don't get enough proteins, your protein intake is low, then you have more energy intake foods, but that quality is low, then what happens, you will get this sarcopenic disease. Sarcopenic disease, that is a sarcopenic obesity, of which you develop the obesity that is related to energy intake, because you don't have enough proteins, no protein synthesis that's going on. So you need much more proteins in your body at the older ages, but not at the young ages. So in that case, body composition changes and the hormonal system changes, metabolic changes, you become obesic. In that case, your aging process becomes very faster because you don't have this protein that has to maintain your body, maybe healing and creating maybe new cellular generation. You don't have this because no protein synthesis. And therefore, we need to understand what is happening in our body. We need to have more proteins at the older age, but not in the younger age. But the problem is, how much do we activate our immune system? How much do we activate our mTOR? We need to get a balance. How do we balance this one? So scientists may be telling us what to do next. So our aim here was to understand what are the mechanisms that are influencing the growth and the performance of the organism. What are the physiological mechanisms? Here, we found that nutrients are activating the mTORs, especially amino acids that are responsible to activate the mTOR pathway. So we selected methionine because it's the first limiting amino acid. It's found in the world, but it's in a very limited amount. And it's responsible to activate the mTOR pathway, and it's a building block in protein synthesis necessary for normal growth and development. And in that case, it's involved in metabolic pathway that is synthesis of this antioxidant glutathione production. And leucine the same, apart from being the signaling molecule of and the building blocks of the proteins, still is a major mTOR signaling pathway. That's why we have selected this one as one of our amino acid to investigate what's happening in birds. So what we did, we used this amino acid. We bought this one from the suppliers and made a solution which is almost 1 milligram per egg, and injected this one in eggs, 1 milligram per egg. And we had the two groups. And then what we did, that was done before incubation. Then we incubated the eggs under standard conditions. So we injected this by using a Hamilton syringe. Incubated under standard conditions of the incubator, and after hatching, we took some blood samples immediately, day one, and sacrificed some birds and collected some blood and tissue samples, but some of them were reared until they reached 21 days in this cage. And this was under standard conditions that was prescribed by other studies. We had some blood samples that we collected by using these capillary tubes and investigated the laboratory, we used this ELISA, and we did some of the liver sample collection and stored in the -80, and then we did PCR, post-protein chain reaction, by isolation of the RNA and DNA synthesis. Finally, we got the qPCR product, but we ruptured the cells by using mechanical method, by using this rotor. And later, we analyzed our data by using the linear model method and the linear mixed model, and then estimated margin mean to compare the body mass and morphological traits. What we found in our results that there was an increase in body mass from day 7 for the beds that were injected with the amino acids initially towards day 21. Those beds that we kept them for quite a long time, at least to 21 days. So we found that there is a difference. The difference was seen from day 7 to day 21 of the methionine injected group as compared to the control group. And we also found that IGF-1 was much more expressed in 21 days old chicks as compared to day 1 old chicks. And also we found some genes were regulated earlier than the other genes. On day 1, we found the IGF-1 was much more expressed in chicks that were injected with methionine as compared to day 21. That means this IGF-1 was at least equal, phased out, that didn't influence, the methionine didn't influence on day 21. But it turned mTOR was high in methionine injected during day 1, but it's opposed to day 21. However, ribosomal protein, serine 6 kinase, was highly regulated in day 21 old chicks compared to day 1 old chicks. This was like a lab transition that some genes were regulated in the early stages of development, but some genes in later stages of development. Unfortunately, we did not find some differences in morphological traits. We measured the wing length, the head length, and tarsal length. In this case, there was no much more allocation to these morphological traits. So the birds were allocating more resources to body mass as compared to the morphological traits. We also used the amino acids as one of our nutrients. We mentioned this one, how does it do? But in this case, we incubated a small amount of eggs. But under this case, we tried to check out if we inject it in day 0, embryonic day 0 and then day 10. So this is day 0, this should be day 0. And embryonic day, that's before incubation, and this is day 10, that's after incubation, day 10 days after incubation. So there are two experimental manipulations here. Day 0, before incubation, and day 10 after incubation. What we found here, these are different results. So for day 0, unfortunately here, we did not get, maybe due to technical errors, that we did not get enough chicks that were raised up until 21 days. In day 0 injection, due to technical issues, but few chicks hatched. So we compared if we inject it on day 0 and day 10, what results do we get here in terms of body mass? We found that there was no significant difference on the day old chicks. There was no effect. But we also found that if you inject it in day 10, this is day 10, and then we kept them and some of the birds until 21 days. At least this part here, this is like the significant difference for the body mass compared to the other. I mean, if you compare this to day 0 of the methionine, this gives a best result in methionine, but it's not comparable because days of injection is different. And Also, we found that in terms of head length, there was no influence between day 0 and day 10, but there was a significant difference in embryonic day 10 increased body mass changes in all the leucine-injected individuals. And tarsal lines followed the same trend, but this one began in day 3 towards day 21. But day 0 and day 10 had no significant in terms of tarsal length. The same applied to the wing length. We found that wing length was significant from day 10, but this is for day 10, this increased from day 7. And also we measured the IGF-1 levels in this 0 and day 10 injection. What we found that IGF-1 increased in leucine injected on day 0, and day 10 at least we found that yes, there's a little bit increase, but the significance was not, the level of the difference was not that significant. And also in day 10, we found that day-old chicks had no difference, but there was a little increase in day 21 of the IGF-1. And after that, we sacrificed some chicks on day 21, and on day 1, we collected intestine, and we measured what's happened within the intestine. And we found that those chicks that we injected day 10 amino acid, the difference was high compared to non-injected, and there was no difference in day 0 and day 10. So what do we get here? What the message do we get here is that a very subtle increase, a little increase in specific amino acids on the inset of embryonic development has got an influence on some genes. That some genes are regulated in the early stages and some genes regulated later in life. So it resulted into a significant increase in growth and after some time, but in methionine, for example, after 3 weeks later, then some genes also you find that they are regulated. For example, the ribosomal serine 6 kinase was found that it's highly regulated in 21 days old chicks. And it's clear that genes and some hormones are regulated in different time points, depending on the way they respond to these molecular pathways. So offsprings usually grow by sensing the maternal effects. We injected amino acids to reflect back. Maybe if amino acid is increased, is more allocated to the eggs, what could happen? So the reflection is that the magnitude of the maternal effects, maternal specific nutrients, once allocated to the individuals, to the eggs, in the early stage of development, during this sensitive window of development, there is a chance that this effect will be progressing towards the post-natal development. So maternal derived nutrients cues may be powerful programming effects on postnatal development, but indeed these changes actually are important during the early stage of development, so they may influence the phenotypic development later in time. But we could ask ourselves, what amount of amino acids do we need actually? For example, we human beings, what amount of amino acids do we need in order to grow? We have seen that if you eat more amino acids or more proteins, the mTOR pathway is being activated. So if mTOR pathway is activated, what is the impact? That there is a chance that there will be a development of diseases like the cancer that we have already seen, sarcopenia. We can reduce this by reducing the intake of some of the specific amino acids or proteins that are activating the amino activating the mTOR pathway. So we need to reduce the amount of food so that we cannot influence the activation of the mTOR pathway, but we need to strike into balance. What amount of amino acid do we need? How much? To what extent should we use the amino acids? In this case, everyone in his or her life should understand what I need. Maybe later in time, in young stages, yes, you need it for development, but you don't need it too much. It should be a little bit quite low, but during the later in life, you need it for at least to maintain the balance between the energy intake and the proper nutrient intake, specifically proteins, at least to prevent this sarcopenic obesity that develops during the elderly stage. So we have done some of the other activities, like we investigated how stress response are related to, but the results are not shown here. Stress response are related to the age of the organisms. Maybe we will show it later on. We tried to check out to induce stress in beds, and in those beds were at different ages. Then we found that there are some differences that organisms respond in terms of stress, but because they are not connected to these nutrients, so maybe we should later on in the same beds, but not in terms of nutrients, but just a stress response. So what next? We are going to investigate how amino acids manipulation at different age groups. For example, if you give amino acids at the lower age of development, at the lower stage of development, what could be the influence in the nutrient sensing pathway? What could be the response in the nutrient sensing pathways? Then maybe at the middle stage and the later in life, what could be the response in terms of this phenotypic expression? What would be the response? How would they influence this mTOR pathway instead? So we expect maybe if we could get enough resources, then we do this. So I will come in, maybe you can have some inputs. This could take up the end of my presentation, and thank you very much for coming.
Thank you. Questions?
I have two questions. Maybe both of them are stupid, but did you have any deaths? Any of your eggs died during the experiment, or all of the eggs hatched, all the chicks hatched in the experiment?
Yeah, during the embryonic development, we actually scanned the eggs by using light. Then we found that some embryos died at a certain stage, but we couldn't manage to understand at which stage these died, because we scanned the eggs at day 10 after incubation. And then we found that some embryos started to develop and then died somewhere. Mm-hmm. The reason could be maybe one is either the injection could have disturbed the makeup of the egg, the organization of the egg itself, the materials in the egg, and two, maybe the nutrients that were deposited in the egg initially was not enough to develop to its final state. Mm-hmm. Yeah.
And did you have any difference between the control and the treatment groups in terms of dying?
Yeah, exactly. We found that there's a difference. Those that were injected, of course, they hatched low compared to the control group. Mm-hmm. I guess injection itself could have an influence, maybe, but the results are significant in terms of the response to amino acids in the injected group. So injecting cells, because it's not natural, it could have disturbed the organization.
So it might be then that what effect you found is not the effect of the injection of the amino acids. It might be a selection in chicks. So basically, you give these injections and you kill some of the chicks, and only the remaining chicks, which might, just because they actually has a higher something you measure. So, make it sense?
I'm not so much convinced with maybe the injection could have killed. Maybe it could be the toxicity of the amino acid itself, but not the injection. If it's an increase in amino acid, that means obviously mTOR activation becomes higher. If mTOR activation becomes higher, there is a problem that multiplication of cell division, cellular proliferation is becoming higher, and this is a problem that we have already talked about this. If there is higher increase or higher activation of the mTOR pathway, it becomes a problem. So it is the possibility that amino acids themselves became maybe toxic, because what was increased just maybe 1%, some embryos could have not been responded properly to the increase of the amino acids. So this could be the problem.
No one else has questions, so I have another one. But biologically, how relevant to inject eggs after 10 days of incubation? Because that can never happen in nature.
Yeah, exactly. That could never happen. But in terms of production point of view, yes, it can. It is done, and mostly in chicken, it's done. But we wanted to see what is the response if it happens that because during development there is creation of the space inside the egg, because embryos do absorb some of the materials in the egg. So there's an empty space, and most of the nutrients are being absorbed at the final 3 days before hatching. So in that case, we wanted to see what is the difference, at least to this leucine injection. What could be, can you get a difference? Of course, there is a reflection of some kind of, for example, hormones, but we are waiting for the other result because we have not yet investigated how mTOR pathway responds to data injection. We still have not those results. We are waiting for some of the important materials to arrive. Then we can do the laboratory work. We arrive with the results on data injection, how it responded to mTOR pathway. Maybe we have some clue on. But in production point of view, of course, yes, it increases also hatchability. Those animal scientists, they do that most of the time, at least to increase the growth rate of the animals. Because in the end, what is required actually is the growth rate of the animal. And we have seen here that growth rate was increasing in the methionine injected groups. And this is what animal scientists, in terms of production, they want to increase the body mass of the broilers. So they need to increase the weight. So to do that, at least they have to supplement some amino acids, maybe during some stages of development.
So if the mass increased in the treatment group, but the tarsus length and the head length and the other length measurements didn't have any difference, where does the extra mass comes from? More fat or more muscles, or what do you think?
Yeah, thank you for the next question. Basically, if mTOR is activated, what is the repercussion behind? We get high protein synthesis. So most of the proteins are deposited in muscles. So muscle mass increases. In this case, the feather length, tarsal length is just the skeletal.
Mm.
So the probability that skeletal increase was not there, instead there was an increase in package of the muscle mass instead of the skeletal system. So protein synthesis was high, therefore most of the proteins were deposited in the muscles. That's why there was an increase in the body mass as compared to the skeletal system, in which most of the cases calcium is packed into the skeletal system instead of proteins.
No.
Yeah.
So in muscles. Okay. And another question I have is, all the eggs were the first egg of the mother, or were maybe the second or third in the clutch or something? I don't know if it's clear with the question.
We are not so sure because
So,
we had several beds that we are laying eggs. And then we collected them. The principle that you usually collected them within 5 days. So we are not so sure whether it was the first egg or second egg, but the collection should be within 5 days. So we couldn't manage to understand whether this is a first egg or second egg, and they were laying continuously. Yeah.
so because I'm thinking that, well, I think it's known that the more eggs the mother lays, right? Well, probably, or well, it maybe can be safe to assume that the first eggs maybe have more nutrients than the others, or maybe that they can even like allocate differently the resources depending on the number of the eggs. So what if the number of the eggs in the clutch can influence actually the results?
Yeah, exactly. I understand that. Yeah, there's an influence on hatching. One is hatching asynchrony, and also the development of the eggs. But because we kept them in a similar environment, and they kept it continuously laying, maybe I may assure myself that there was no hatching asynchrony. Otherwise, maybe due to age difference. But our beds were of the same age. They were obtained from the same generation. So I would say that if there is hatching asynchrony, maybe all growth asynchrony could be maybe balanced in terms of maybe growth or whatever. But I wouldn't have a proper answer for that. If we could have maybe that whether if this is first egg or second egg, because we didn't check out.
Can you have another question?
Let's second.
And I mean, I'm just curious, like, why choose this experimental or this approach? So, why is it different if you, for example, would just feed a group of mothers with a methionine-induced diet, or a diet with a higher amino acid, and then see the effect on the embryos, or like that? So
yeah, exactly. That's another suggestion of the experimental approach that it could be. But at least we wanted to approach this directly. Maybe we can just imitate or reflect back. What if it happens that there is an increase? What if it happens there is an increase in allocation to the mothers? But that's a different approach. That if you feed to the mother and then investigate on the chick, that's transgenerational maternal effect. But we wanted to reflect how if it happens, there is more allocation of specific amino acids to the eggs. So we approach it in this way, but not by feeding the mothers. But that's possible also, and some of the experiments maybe in poultry they've done that, but not to the specific amino acid. Maybe just feeding the nutrients and check out what is the response to in terms of phenotypic expression of the offspring But maybe we can consider later on if we can have a possibility to do that. We can do that also.
Thank you very much. If there are no more questions, then I thank Sawadi again for the talk, and I have a very bad news because Sawadi tried to convince us in order to extend our lifespan, we should eat less, and after careful consideration, I reject this advice, and what I advise is that we go and shorten our lifespan a little bit by eating some lunch together. So thanks a lot again.